Today what we're going to be talking about is how to take a human cell count in a biochemistry lab. The purpose of this lab video is to show how to take a human cell count, show how small human cells are, and demonstrate the work that is being done every day in a biochemistry lab. It is important to remember throughout this whole process, safety and sanitation is taken into effect. So the first step of this process is to remove the media by aspiration. This occurs after the test tube comes out of a centrifuge, and it is important to remember that the tip of the aspiration tube should just be at the top of the liquid and move down as the media is being removed. At the end of this process, the only thing in that test tube should be the human cells. Immediately after this is done, 1 milliliter of fresh media needs to be placed back into the test tube. This prevents the cells from drying out. After the fresh media is placed, the fresh media and cells need to be mixed together to form a homogeneous mixture. Once that mixture is produced, 10 microliters of that cell suspension needs to be added to 10 microliters of trypan blue. It's important to already have that trypan blue measured out and placed into its own vial. After the trypan blue and the cell suspension has been created, take 10 microliters of that particular solution and place it into a slide. This slide then needs to be placed into an automated cell counter. Wait for the reading on the cell counter and record the total cell count, the live cell count, and the live cell percentage. Discard that slide into the biohazard waste receptacle. Little check on learning here. Which stain was used to mix with the cell suspension for this process? Answer is trypan blue. Little closing remarks here is just a summary of the 8 steps that were used for this particular process.